All right, so part one, we're going to deal with the situation of two genes being on the same chromosome. So we have two genes, one we call A, one we call B, and then each one of these has two alleles, so big A and little a. So for this chapter, you can assume dominance, recessiveness apply. So let's say you have a heterozygote individual. So this individual is big A, big B, little b. And so when an individual with this genotype, and these two genes are in different chromosomes, this is kind of like the end of the situation. But when two genes are on the same chromosome, there's actually two different scenarios that can lead to the creation of a heterozygote individual. And so this is what's shown on the left and on the right. And so in both cases, you have a sperm fertilizing an egg. The sperm is a haploid, the egg is a haploid, but there's actually two different situations could lead to an individual having the same genotype. So in the first case, the father, the homologous chromosome from the father, has dominant alleles of both. The mother has recessive alleles of both. And so when we combine these, when fertilization occurs, you end up with a genotype with big A, big B, little b. In the second scenario, the haploid from the father, the homologous chromosome from the father, contains one dominant allele. So A is dominant and B is recessive. And then the mother's haploid cell has the opposite. So it has the recessive of A and the dominant of B. So again, when you combine these to fertilize an egg together, you result with a genotype of big A, big B, little b. However, the parental gametes, the original combinations, are different. And so the parental gametes are listed here, big A, big B, and little b versus big A, little b, little a, big B. And so this motivates us to actually have a new way to write out the genotype of an individual to also give this information. So if you see something like this, this is why we would write it in this way. So big A, big B, little b. So basically that is the parental combinations of the alleles. And so you can see this just simply matches this and this matches this, whereas in this case, this matches this and this matches this. So if you see something written like this, one of the things you should assume is that I'm dealing with a situation where the two genes are on the same chromosome, and I'm providing extra information about the original parental combination of these alleles. So you are an individual that is heterozygote, and now we're interested in the gametes that can be produced by that individual. So this individual right here, what we're showing is the scenario one, because one of the parents had both dominant alleles and the other parent had both recessive alleles. And so you can see this by the original parental combination, the blue being little a, little b, and the orange being big A, big B. So if crossover didn't occur during prophase 1, the only types of gametes that could be produced by an individual are big A, big B, or little a, little b. But because crossover occurs, we now can also produce these new combinations. And so we have the parental gametes, which are also sometimes called the non-crossover gametes that can be produced. But now we can also have these new combinations that get produced because of these chimeric chromosomes that get produced during meiosis 1. So because there's this crossover, we have a situation where we initially have a chromosome from the father, and then it moves into the chromosome from the mother. And then the vice versa is also created. And so when we look at the gametes that are produced, now we can have all 4 different ones that get produced. So again, just to kind of summarize this, so if 2 genes are found on different chromosomes, then we have 50% of the original parental combination and 50% of the new recombinant gametes, the new combinations. If we have 100% linkage, so if crossover doesn't occur, then the only types of gametes that this individual can produce are the parental gametes. And then C, when 2 genes are on the same pair of chromosomes, exchange can occur. However, this is not at an equal 25%, 25%, 25%, 25% probability. So as a result, the parental gametes are the most likely to have, the parental combinations are the most likely to be found in the gametes, somewhere between 50 and 100%. And then the recombinant gametes are somewhere in between 0 to 50%. And so why is that? Why? Because in order for you to have a recombinant gamete, the crossover must occur in between the genes. And so the only way this individual can produce a crossover is if the only way this individual can produce these recombinant gametes is if the crossover occurs in between these two genes. And so the larger the distance between the two genes, the more likely that something like that happens. And so the physical distance is simply the number of nucleotides between two genes that is correlated with the genetic distance, which is the probability that recombinant gametes form. So this is what motivates a unit of measure for the genetic distance between two genes. And that's either called a map unit, or it's also often called a centimorgan for one of the original discoverers of linkage. So 1% recombination between two genes on a chromosome. So these are relative different distances, not exact ones. And in general, 50 map units is the size of an entire chromosome. So the largest size two genes can be apart is 50 map units apart. So how do you calculate this? The way you calculate this is that you have to simply calculate the number of recombinant gametes that are produced by an individual and the number of parental gametes that are produced by an individual. So in scenario one, linkage, two genes are unlinked. So the genotype is simply, heterozygote, big A, little a, big B, little b. And then the gametes that are produced, 1/4 of each type is produced. And so the percent of recombinant gametes is 50%. And so when genes are on different chromosomes, you don't typically report a map distance. It only makes sense if genes are physically linked together. So scenario 2 is the case where two genes are on the same chromosome. And so the genotype that we have for this individual is big A, big B over little a, little b. So of course that means that one parental combination is both dominant, one parental combination is both recessive. And so if we look at those offspring and we look at the number of gametes that produced, and we found, just empirically, we found these numbers, this is certainly a possible thing that we can find. So for this individual, we find that 3/8 of the gametes are big A, big B, 3/8 are little a, little b, and then the other two are 1/8 and 1/8. So the number of parental gametes that are formed, so 3/8 plus 3/8 is 3/4, so 75% of the gametes are parental. And then if we add up the recombinant gametes, 1/8 and 1/8, 25% are recombinant gametes. So to do these problems, you have to become very comfortable with identifying parental gametes and recombinant gametes. And so the percentage of recombinant gametes is equal to 25%. And so this is simply how we measure the map distance between two genes. So 25 centimorgans is the same as 25%. And so that is our unit of measure of how far these two genes are apart. So they're 25 centimorgans apart. So here's another example. So big A, big B, little b is a heterozygote animal. And we're interested in calculating the map distance between A and B. So how do we do this? So now we're getting into kind of like the experiment that we might perform in order to calculate this. So to calculate the genetic distance, what we really need to know is how many of each of the gametes are produced. Okay. So these are the 4 gametes, and this is what we want to know. So we can take an individual, let's say we have a fly, a male fly, and we can get this individual and, we can try to figure out, what the genotype of the gamete is. So the idea is that this fly produces a large number of sperm cells, and each of these sperm cells contains a different content of DNA. In general, however, experimentally, it's almost impossible to figure out the genotype of a sperm cell. So it's not something that we can simply just say we're going to sequence this cell and figure out what the allele is, what alleles it's carrying. So this is what motivates us to do this test cross. So this test cross simply allows us to infer what are the gametes that are produced by this individual. So we cross it to this heterozygote recessive individual, just to remind you what a test cross is. And this individual can only produce little a, little b. So this individual has thousands. So this cross produces thousands, let's say 1,000 offspring. And now we can measure the phenotype of the offspring very easily. So it's very easy to measure the phenotype of an offspring. So we can look at the color of the eyes of the fly, or we can measure, does the fly have wings or not? There's all sorts of different phenotypes that you can measure of a fly. And so we can count of those 1,000 offspring, what is the phenotype of the offspring. And so from that, we can infer what the genotype of the offspring is. And so just as a reminder, for a test cross, we always get one recessive allele of each gene. And so if the individual has the dominant phenotype of both traits, then we know that the genotype is heterozygote. If the individual is dominant for one, then we know this is the genotype. Dominant for the other, this is the genotype, and recessive for both, and this is the genotype. So we know for a fact that one of the alleles is always the recessive because of the way we design the cross. So if this is the genotype, then we can infer what the first parent's gamete is. So if this is, heterozygote, since we know parent 2 is always this, then we can basically just infer what are the gametes. So this is basically how we've inferred what percentage of gametes are produced by this individual. So 400 of these individuals were found, 100 of these individuals, 100 and 400. So the first thing we need to do is look over this table and infer what are the parental gametes. So what was the original combination? And so to do that, we simply look at what are the most common. So because there's way more of the parental, then we know that the original combination of this individual was this and this. So one of the things we can do, for example, is rewrite the genotype of that individual to something like this. And so that just lets us know that this is the original combinations of the alleles that were inherited from their parents. And then the second thing we can do is calculate the map distance between these two genes. And so we count the number of recombinant gametes, 200, and we divide by the total number of gametes that were produced. So this is a very simple calculation because there's an even number of animals. There's 1,000 animals. And so that gets us to 20%, or 20 centimorgans, or 20 map units. So very straightforward. You're always thinking about the gametes that are produced by the individual. So this is one of the reasons in Chapter 3 and Chapter 4 we focus so much on what gametes get produced by an individual, is because it's necessary. You really need to think this way in order to be able to answer questions for this particular chapter. So hopefully this is all straightforward for you by now. So single crossovers. So these occur between two non-sister chromatids. And so when you have a recombinant, this is something that it looks like. So this is the tetrad. The tetrad has formed where the homologous chromosomes have paired together and then crossover occurs. So how do you figure out what results from this crossover? So if it's a single crossover, like what is depicted right here, then you simply follow this chromosome from the beginning, and then wherever there's a crossover, you just follow to cross over to the other chromosome. So this one right here matches that situation. And then the other one that is produced, you start from here, and then you follow over to here as well. And so this guy right here would create this gamete right here. This top chromatid, because it does not have a crossover, it just simply goes all the way to the end. And so this is one of the non-crossover gametes that get produced. And then similarly on the bottom, there's a gamete that would be produced that results from a non-crossover as well. You can also have double crossovers. So in this situation, we have two different exchanges that have occurred between the same chromatids. And so what kind of gametes results from this? So you just follow the same play. So you start from, one of these gametes right here, and then you move till you reach the crossover. You go to the other homologous chromosome, you draw that, and then you reach another crossover, and then you move over to the other side. And so this one right here would produce this type of crossover. So you'd initially have mother's chromosome, then you would have father's, and then you would have mother's. And then the opposite would occur right here. So you'd start here, you would cross over, you'd go onto this chromosome right here, and then you would cross back. And so this double crossover creates blue, orange, blue as a result of it. And then here we have some non-crossovers as well. Okay. So hopefully this is straightforward, and you can actually have triple crossovers, quadruple crossovers, and each one becomes less and less likely. So the single crossover is most likely, the double crossover is the next most likely, and then the triple crossover, et cetera, et cetera. So in that situation for the triple crossover, you'd simply have one more location where it occurred. Maybe it's right here. And so then you just kind of keep following this procedure to kind of go back and forth to figure out what is created. So double crossovers become very important for the next part of the chapter when we start to look at 3 genes. And so this hopefully starts to help you understand that, because you have situations where, so the parental combination is big A, big B, big C. So you have gametes that are created by these double crossovers where the A and the C are the original parental combination, but the B, the middle gene, is actually flipped to the other allele. So finally, what happens if I ask you for crosses between multiple heterozygotes? So what happens in an intercross between individuals of this? So AB, little ab. So what happens if we cross 2 individuals together with the same genotype? What is the offspring that would be produced? And so Punnett square, hopefully by this time, you become very comfortable with. Again, the Punnett square is set up to answer this question very straightforwardly. So the top of this Punnett square are the gametes that are produced by one individual. So this individual right here. The side are the gametes that are produced by this individual right here, okay? So we are told that the map unit, the distance between these 2 genes is 20 centimorgans apart. So first thing we need to do are list the 4 gametes that can be produced by this individual, and also the 4 gametes that can be produced by the other individual. So all 4 can be produced. So then we have to recognize what are the recombinant gametes. So the recombinant gametes are big A, little a, big B. And because the map distance is 20 centimorgans apart, then we know that a total of 20% of the gametes that are produced are one of these 2. And so we simply just split it up. So 0.1 and 0.1, and you add that together, that's 20%. So that's kind of where we got this number right here and this number right here. And then all of the gametes probability have to add up to 1, and so we add a 0.4 here and a 0.4 here. So here what we've done is we've simply just, these are the 4 gametes, and this is the probability that each gamete is produced. And then the same is true for the second individual as well, because it's the same genotype. So 40%, 10%, 10%, 40%. And now to fill in the middle of the Punnett square, all we have to do is multiply these numbers together and then join the gametes together. So here we've done that. So 0.4 times 0.4 is 0.16. And then big A, big B is the resulting genotype. And so we can do the same thing for the entirety of the Punnett square. So if I ask you, for example, what is the probability that big A, big B is produced? The answer to that is simply this first part of the square right here, because that's the only square that has that genotype. So the answer is 16% of the animals would have that. Same thing with the recessive. So if you asked how many are homozygous recessive, the answer would be 0.16. And then you can also ask, how many of this genotype? And so we would group these two squares together because they would have the same one. So 0.08 would have this genotype. So you just kind of follow the approach of the previous chapters where you group the similar genotypes, or perhaps you're interested in the same phenotypes. So in that case, you would also have to group all of the similar phenotypes together as well. So for example, this genotype has the same phenotype as this one. So it's all hopefully very straightforward for you from here.